In any event, I apologize for that, and I had every intention of doing it, but at the end of the term, things start piling up, and they definitely piled up on me. So if you're around this summer, come give me a holler, and maybe we'll go out for a beer for that. How about that? I know some of you are around. So in any event, there's an ant on the floor. There's been an ant on the floor the past 2 days, and I've been very tempted to just squash it, but I haven't done it. And I'll probably squash it as I'm walking around, but it's I don't know if it's the same ant or not, but it's been walking around here for a couple of days. In any event, we have topic of photosynthesis, and we have the topic of nitrogen metabolism to cover in the time that we have remaining. And so I will get through these. Photosynthesis, as I noted with you yesterday with the photosynthetic fish, is a fun thing to think about, and realizing that everything that we know of on Earth ultimately happens because of photosynthesis. So photosynthesis is a really remarkable process. All of the biological energy that we see on the face of the Earth ultimately comes from photosynthesis, at least as far as we know. Okay? So that's a pretty cool thing. I showed you chloroplasts yesterday, and I want to say a little bit about what's happening in the process of photosynthesis. And I'm sure you've had some of this in your biology classes before, or your botany plant pathology classes before, but I'll say a little bit more here. And we'll see it a little bit more up close and personal than you've been seeing it. But photosynthesis is a process that occurs in 2 cycles. You refer to them as the light cycle and the dark cycle. And the light cycle is something that's completely dependent upon light. The dark cycle, however, is not dependent upon light. It's not dependent upon dark either. Okay? So it's called the dark cycle because it can occur in the dark, but it is not something that requires darkness. So the dark cycle is probably going on during the light cycle as well. Okay? Now, as I noted yesterday, in photosynthesis, what's happening is some processes very similar to what we see happening in electron transport and oxidative phosphorylation in the mitochondria. But instead of protons being pumped out of the mitochondria and into the intermembrane space, we see protons being pumped out of the stroma of the mitochondrion, which is this sort of central space in here, and into the thylakoid spaces that we will see. Okay? So instead of going out of a structure, it's going into a structure. And as I noted yesterday, that gradient that happens still is useful. It's just occurring in the opposite direction. And that gradient is still useful for making ATP. Okay? So the light, in order to do something with light, you have to be able to capture the energy of light. And that's the function of the chlorophyll, which we think of course, as the green causing substance that's in the chloroplast. And if you remember your structures of heme, which I'm sure you all do very well, you'll note that the structure of chlorophyll is very, very similar to that of heme that's found in our blood cells, with a major difference. And the major difference is that instead of having an iron at its center, the the chlorophyll actually has a magnesium at its center. Okay? You notice that this has a long hydrophobic side chain coming off of it, and that helps to anchor the chlorophyll into the thylakoid membrane within the chloroplast. Okay? And what happens in photosynthesis is something that's interesting and it's not completely understood, but it is known that a single photon of light, of course, can excite electrons, as we shall see, and that energy that comes from that photon of light can actually be transmitted across across several chlorophylls, as you can see here. So that energy is not something that's trapped to one place, but actually does travel, and it's not completely understood how that traveling actually happens. What I want to do is take you through this process that you can see on the screen, and I show you all this not to give you a whole bunch of different names of molecules to memorize, because I don't think that's important. But I will talk to you about some of the highlights of what's happening here. Okay? Now, this process is really interesting, and as I'm going to show you, in some ways it is occurring in a sort of backwards fashion compared to electron transport. It's a sort of a backwards fashion. Now, you won't see that, you won't really understand that until I get you pretty much most of the way through the process. But for the process of photosynthesis, okay, the overall process starts in something called Photosystem II. Okay? So there are two photosystems, Photosystem I and Photosystem II, and they were numbered before they realized exactly how they were working. But the process starts in Photosystem II. And this process happens with the absorption of a photon of light by a chlorophyll. Okay? There are complexes within the chloroplast that have the ability to, we can think within the chlorophyll, the ability to lose electrons. Okay? So when something absorbs a photon, or when you see light or anything like that, there's something happening to the electronic structure of a molecule. And in this case, the photon interacting with the chlorophyll causes an electron within that molecule to get excited. And it gets excited in such a sense that it actually leaves the molecule. Okay? So it's getting excited and leaving the molecule. That causes the molecule that's left behind, this P680 molecule that's left behind, it's lacking an electron, and it really, really wants an electron. Okay? It really, really wants an electron. So how does it get electrons? Well, it turns out that the way that it gets electrons is it has an electron donor. And the donor, it actually physically, and it's not really what I would think of as a donor. It's more like it's actually being a bully and taking the electron. It's actually taking two electrons, but for that matter, it doesn't matter for our purposes here. It's taking an electron from water. This has the effect of oxidizing water. Now, oxidizing water is something that people are very, very interested in doing because, A, we have a hell of a lot of water on this planet, and B, it's not readily oxidized. In fact, if you think about it, when we put a match to water, the water puts the match out, right? So oxidizing water is pretty exciting. So when you see people working on various components of biotechnology and bioenergy, one of the things that they're really interested in is what's actually happening right here. So water gets oxidized. The oxidation of water produces oxygen, okay? And it produces protons. So one of the things that the first things that you should remember about photosynthesis is it is an oxygen-generating process. It is an oxygen-generating process. Okay? We know that the oxygen that's in our atmosphere today is here because over the billions of years that we've had the process of photosynthesis going on, there's been enough oxygen liberated by this process right here to give us what we have in our atmosphere. We know that the original atmosphere of Earth had very, very, very little oxygen in it. So we have the oxygen in our atmosphere thanks to this process that you see on the screen. Okay? That's pretty cool. We have excited an electron. All right? In the mitochondrion, we didn't have to excite an electron. Why didn't we have to excite an electron? Because we already had an electron at a high energy state in NADH or FADH2. So this process is starting with an excitation. The excitation removes an electron from the system, and that system takes away oxygen from, takes an electron away from water, making oxygen, sorry. Well, what happens to that electron that got taken away? Well, this is a very high energy electron, or pair of electrons. And these electrons get passed through a series of electron carriers. And these electron carriers are in the thylakoid membrane of the chloroplast. So that little flattened disc structure that I showed you yesterday is the thylakoid. The membrane of that is where these electron carriers are found. Well, you've seen what happens in electron transport in our mitochondria. Complex 1 donates to coenzyme Q, donates to complex 3, donates to cytochrome C, donates to complex 4. And so we see the movement of electrons through a variety of carriers in the thylakoid membrane, just like we saw in the mitochondrion for electron transport. So this process now is very much mirroring what's happening in electron transport. And in electron transport, we saw that the movement of electrons through some of the complexes caused protons to be pumped out of the mitochondrion and into the intermembrane space. Okay? In this case, we see pumping of protons, but again, the protons are being pumped not out of the thylakoid space, but into the thylakoid space. So they're taking protons from the stroma of the chloroplast and pumping them into the thylakoid space. This is creating a proton gradient. Okay? And we see the electrons traveling, traveling, traveling. Now we see something different that happens next that didn't happen in the electron transport system in the mitochondria. And that is that the electrons arrive at another photosystem. When they arrive at the other photosystem, you'll notice that the way this is drawn is that the energy of the electron is decreasing, 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 just like water or a ball would roll down a hill. Okay? So the energy of these electrons is decreasing as they're passing through these electron carriers. They get to this photosystem, and this photosystem is waiting for another photon of light. Another photon of light hits this chlorophyll and causes these electrons to get excited again. So it's a two-excitation step that's happening in the process of photosynthesis. The electrons get excited, and they pass through a series of what are called iron-sulfur proteins. They're proteins that contain iron with sulfur. And by the way, we have iron-sulfur proteins in our complexes, in our mitochondria as well. I just didn't tell you about them. They're not that different from what we have. The electrons pass through there, and they pass through this iron-containing complex called ferredoxin. Ferra meaning iron. Okay. Ferredoxin passes electrons off to an enzyme called NADP reductase. Well, a reductase is something that is going to reduce something else. And we have electrons coming, right? So those electrons are going to pass to NADP along with a couple of protons and create NADPH. Now, I mentioned to you that this process is somewhat the reverse of electron transport. Why is that? Well, in electron transport, we started with an electron carrier, NADH, right? Here, we're ending with an electron carrier, NADPH. We've made a carrier that has electrons. Okay? In electron transport, the electron acceptor at the end was oxygen and made water. This process is starting with water and making oxygen. Okay? So how is it possible that we're able to reverse what's happening inside of electron transport? The answer to that is contained right there in those photons of light. The photons of light provide the energy to push the water back up the hill. All right? The water back up the hill corresponds to these electrons that are moving through these complexes and ending up back on NADP to make NADPH. This is what is the sum of what we call the light cycle of photosynthesis. This process absolutely requires light. Absolutely requires light. Questions about what I've just told you? You've just learned the light cycle of photosynthesis. Aren't you guys excited? Yes, question. Any words that I've used here you should know. Okay. So, would you like me to name these off for you? Are you sure? Because that way you'd have an opportunity to learn them, right? I didn't name any of those names in there. Okay? Other questions? Quiet group today. Okay. At the membrane level, this is what's going on. And it's really no different than what I've just told you in that last diagram. That last diagram, by the way, has a name. It's called the Z-scheme because you see up, down, and then back up again. They call that a Z-scheme. All right? But it's really not, I mean, it's happening inside of a membrane just like what you saw before. But notice that in this case, the protons are being moved in, all right? And they flow back out. I haven't told you that part, all right? The last part of this process is that this proton gradient that's created passes through an ATP synthase that is almost identical to the one that's in your mitochondria. It has a mushroom shape like the one in your mitochondria. It has a spinning component like the one in your mitochondria. It makes ATP like the one in your mitochondria. And the difference here is that instead of calling it oxidative phosphorylation, we call it photophosphorylation because it's the light energy that's driving the whole process. In our mitochondria, the thing that's driving the process is oxidation, which is why we call it oxidative phosphorylation. Okay? Pretty cool stuff. Okay, so that's what's happening in the light cycle of photosynthesis. There's a molecule whose name I didn't mention. And I'm not going to mention it, but I'm just going to show it to you so you can admire it in its beauty. And this is a molecule that plays a role in the electron transport process. And my only reason for showing it to you is to sort of remind you something that you've been seeing over and over now this term. And that is, if you have a long hydrophobic tail, that long hydrophobic tail can serve to anchor something inside of a membrane. And since you know that this unnamed molecule is found in the thylakoid membrane, you see again the role of this hydrophobic tail in anchoring it inside of there. If anybody remembers the structure of coenzyme Q, this looks very much like coenzyme Q, and it's actually chemically related to it as well. Here's some more. Look at those tails. Okay. Now, plants have some interesting challenges. All right? And it depends a little bit on where they are. We're going to talk about a couple different challenges. The first challenge I'm going to talk to you about is a challenge of having too much light. What if you have too much light? Well, let's think about what we've just learned in the process I've just described to you. What was the terminal electron acceptor? NADP, right? And that made NADPH, right? What was limiting inside of your cell with respect to metabolism? When we talked about glycolysis and the reason we had fermentation, what was limiting? Oxygen was limiting, and oxygen's limitation made for something else being limiting, which was NAD. Cells have limited amounts of electron carriers. A plant that's sitting out in the sun and getting hours and hours and hours of light is going to have a limited amount of electron acceptor. If it doesn't have enough electron acceptor, okay, then what's going to happen to this whole process I've been describing to you? Well, it's not going to be very efficient. It's going to run for a while, and then it's going to wait for the NADP to get used. Okay? Well, it turns out that plants have a sort of short-circuiting that they can do that's called cyclic photophosphorylation that reduces the need to have a terminal electron acceptor. This is different from oxidative phosphorylation. I like to think of this as literally short-circuiting the process. This is occurring in Photosystem I. And we see excitement of electrons by a photon. And we see the short-circuiting of the electrons that are passing through Photosystem I passing back through some other cytochromes that are found in the membrane. And the result of this is that pumping of protons occurs. So this process is kind of like running that other process backwards and short-circuiting it. We're passing electrons from photosystem I back through the proton pumping system to create a proton gradient. Now those electrons remain internal inside of complex 1. They don't go anywhere else. They short-circuit. They literally do short-circuit. Well, the advantage of this is it doesn't need an electron acceptor at the end, because the electrons come right back to the place where they started. But more importantly, protons get pumped as a result of the photons coming in. This system will kick into place when there's no additional NADP. Okay? Well, why is this important? This is important because the cell can continue to make ATP when there's plenty of light but no electron acceptor. Cyclic photophosphorylation is a very important process that occurs inside of plants. And it keeps plants producing ATP all day long when there's no electron carrier. Okay, now, there's the mess that I just showed you, which I'm not going to go back through. We'll bypass that, okay? And instead, I want to not talk about that. What I wanted to talk about here is actually down here. Well, no, actually, okay, I'm going to come back to that. Let me come back to that. All right. I should have looked at this before I came. Damn, okay. All right, so that covers what I want to say for right now about the oxidative photophosphorylation process. I'm going to come back to that in that very last set of reactions and tell you a little bit more about that, but it crosses into the dark cycle of photosynthesis. All right? So we've talked about the light cycle. I want to now turn our attention to the dark cycle of photosynthesis. And as I noted before, the dark cycle does not require darkness. It's going on during the light cycle as well. Okay? And it's important that it go on during the light cycle, because one of the things that the dark cycle is going to do is it's going to use NADPH, and that's going to provide an electron carrier. Photophosphorylation, the cyclic photophosphorylation that I've just described to you, is fairly inefficient. It's most efficient if we go through the regular photophosphorylation that I told you about before. Okay. You guys want to stretch before we do this? You want my duck joke? You want a duck joke? Okay. So, this is a dumb joke, but I'm going to tell you this joke anyway. Okay? That's never stopped me before. There's these three guys, and they go to heaven. Okay? And they get up in heaven, and they get to the pearly gates. And at the pearly gates, they look, and there's these ducks everywhere on the ground. And they get to the pearly gates, and they say, "Welcome to heaven. We're happy to have you here. But whatever you do, don't step on a duck." Anybody heard this joke? You've heard the joke? Okay. Yeah. Don't tell the punchline. Okay. It's a dumb joke, all right? Don't step on a duck. Okay, yeah, we're not going to step on a duck. So they go wandering around heaven, having a good time. And after a day or so, one of them steps on a duck. And all of a sudden, these sirens go off, and these police from heaven, which is kind of a concept in itself, these police from heaven come, and they grab this guy, okay, and say, "You stepped on a duck." And they got this mean old woman. And by the way, this could be three women and men. And it's not I don't mean to make a sexist joke, so it's not a sexist joke. There's three mean old women, and they chain this mean old woman to this guy, and they say, "You stepped on a duck. The two of you are going to be united forever like this." Wow. So these other two guys are pretty blown away, man. I mean, they're going to, you step on a duck, the consequences are pretty severe. So they're very, very careful not to step on a duck, right? They don't step on a duck. And after a couple of weeks, one of them accidentally, not knowing what he's doing, steps on a duck. Same thing happens. They come, and they got this woman, she's got a frying pan. And they chain this guy, and she's beating this guy in the head with a frying pan, saying, speaking of stereotyping here, beats this guy on the head with a frying pan, right? You're going to stay chained to her for eternity because you stepped on a duck. Oh, man. Well, the third guy is absolutely paranoid. He doesn't want to move. He wants to just stand here because he's afraid if he shuffles his feet, if he does anything, he's going to step on a duck and he's going to spend the rest of eternity in misery, right? And a couple of months go by and this guy has not stepped on a duck. Okay? But he knows the consequences. He's not stepping on a duck. And pretty soon the sirens go off. And the police come down, they grab this guy, and he goes, but I didn't step on a duck. And they have this beautiful woman. They chain him to this beautiful woman, and he looks at her and he says, this wasn't what I was expecting. And she says, that's what I get for stepping on a damn duck. Okay. Now that I've gotten that out of my system, that's my last joke of the term. I've told you all the good jokes I know. That's a bad joke, I guess, but anyway. Okay. Now we do the dark cycle. Aren't you glad I got you prepared for the dark cycle? Look at that. Oh, my God. Oh, my God. We're going to go out in style. We're going to go out with a great, brand new cycle of metabolism to learn at the end. Isn't that great? That's what you get for stepping on a duck, guys. No, we're not going to go through this whole thing, all right? In fact, we could. We could go through this whole thing. Am I hearing pain here? Okay. We could go through it, but in fact, many of the reactions that are in what's on the screen, you've already seen. I'd like you to look at some of the intermediates. Dihydroxyacetone phosphate. I would like you to take a look at glucose 6-phosphate, fructose 6-phosphate, ribose 5-phosphate, glyceraldehyde 3-phosphate, 1,3-bisphosphoglycerate, 3-phosphoglycerate. You've seen many of the reactions that are on the screen. And no, you're not going to memorize them in any order. But I want to show you what's happening in the dark cycle of photosynthesis so that you get a sense of it. That's what I want you to have, is a good sense of it and some highlights of it. Okay? First of all, what's happening in the dark cycle of photosynthesis? What's happening in the dark cycle of photosynthesis is carbon dioxide is being pulled out of the atmosphere and made ultimately into glucose. Now, what's confusing to students is they expect to see that the cell is going to take 6 carbon dioxides and put them all together and link them together and add protons and add electrons and make glucose, and that's not what happens. The carbon dioxides are pulled out of the atmosphere one at a time so that through 6 cycles, 1 glucose gets made. Does that make sense? So they're not all happening at once. And you'll see what's happening here. Well, how does this process start? This process starts with a molecule that has 5 carbons. It's a sugar, and it's a sugar called ribulose 1,5-bisphosphate. And yes, that's an important name. You can abbreviate it RUBP if you want to. RUBP, ribulose 1,5-bisphosphate. Okay? The addition of that carbon dioxide, okay, is catalyzed by an enzyme that's very interesting. This enzyme that's very interesting is called ribulose bisphosphate carboxylase, or as you're more likely to call it, Rubisco, R-U-B-I-S-C-O. Rubisco, okay? Rubisco is probably the most abundant enzyme on the face of the earth. Needless to say, from what I've been telling you about all the energy that's on the face of the earth, thanks to photosynthesis, Rubisco is one of the most important enzymes on the face of the earth. The addition of this carbon dioxide to this molecule creates a 6-carbon intermediate that is very unstable. And it falls apart. It falls apart and creates 2 molecules of 3-phosphoglycerate. 2 molecules of 3-phosphoglycerate. Okay? Well, you can start thinking if I've got 3-phosphoglycerate, that's an intermediate in glycolysis, and that's an intermediate in gluconeogenesis, that I could take those molecules and go back and synthesize glucose. And that's largely what's happening here. There's some other intermediates that are happening, blah, blah, blah. But largely what's happening here is that these two molecules here are going back and making a glucose. Okay? 3-phosphoglycerate becomes 1,3-bisphosphoglycerate, blah, blah, blah. Look at the ATP. We need ATP to make that happen. Look at the NADPH. We need NADPH to make the other things happen. So we need energy to make glucose, and we need electrons and protons to make glucose. ATP and NADPH are the products of the light cycle. The light cycle is feeding the dark cycle. Okay? So it's important that the dark cycle be going on while the sun is out, because that's going to produce more NADP, that's going to produce more ATP, and that's going to keep both processes going. Okay? Everything else that's happening in here is balancing the equations. That's really all there is to it. Everything else that's happening in here is balancing the equations. And people get all up in arms about how many of these and how many of these and how many of these so that we balance our equation. And understanding what's going on isn't about balancing equations. It's about seeing the process occurring. When I described to you earlier the pentose phosphate pathway, I described it to you very briefly. And I said that the pentose phosphate pathway has reactions very much like what we see in the Calvin cycle. By the way, this is also called the Calvin cycle. The dark cycle is called the Calvin cycle because that was the person that discovered most of the reactions in it. Okay. The pentose phosphate pathway contained sugars of varying numbers of carbons. I'd like you to look at what's in here. Here's a 4-carbon sugar called erythros 4-phosphate. Here's a 7-carbon sugar called cetoheptulose 1,7-bisphosphate. Okay? There are 6-carbon sugars, fructose 6-phosphate and glucose 6-phosphate. And you know if we get all the way to glucose 6-phosphate, what can happen next? We can make glucose. But glucose is a poison, right? What happens to glucose when the cell makes glucose? Well, in a plant, it makes amylose, it makes amylopectin, and or it makes cellulose. It's got 3 ways of getting rid of glucose. Okay? Questions about this? Okay. Here's that fixation reaction. Fixation is what we call the process of pulling carbon dioxide out of the air. We say that carbon dioxide is fixed, meaning it's linked to something else. And here's the something else it's linked to. No, you don't need to know the name or the structure of that. But you should know that the products are 2 molecules of 3-phosphoglycerate. Those are what people refer to as the first products of the Calvin cycle. The first products of the Calvin cycle. Okay. That's lovely. Okay. Rubisco is, as I said, one of the most abundant enzymes on the face of the earth. And to your surprise, and to the surprise of biochemists who studied it, it's actually a fairly inefficient enzyme. It's fairly inefficient. It doesn't work very well. And when we think about the importance of this enzyme and thinking about that it doesn't work very well, that's a little surprising. Scientists have tinkered with it, trying to make it more efficient so that they can make plants that do more. And the question is, why is it inefficient? That's going to come back up to this last reactions that I'm going to talk about up here. But before I talk about those, I want to talk about another type of plants that exist in the world. How many of you heard of C3 and C4 plants? I'm guessing, oh, most of you have, okay. So, C3 plants are most of the plants on the face of the earth, and they have reactions exactly like what I've just told you. C4 plants are plants that have adapted to relatively dry conditions to conserve water. And this process that they have evolved employs the Calvin cycle that you just saw, but it adds another step to it, or another couple of steps to it. Okay. These are called C4 plants. What's the difference between a C3 plant and a C4 plant? A C3 plant produces, as a result of absorbing carbon dioxide, it produces 2 molecules of 3-phosphoglycerate directly. A C4 plant produces a 4-carbon intermediate as a result of absorbing carbon dioxide directly. It ultimately will produce 2 molecules of 3-phosphoglycerate as well, but not in the first step. The first step involves something else. So, what you see on this is the schematic structure of a C4 plant. And C4 plants, rather than, so what we see on the right is the Calvin cycle that you've already seen in the C3 plant. If this Calvin cycle is placed out here on the edge of this leaf, as it were, okay, these pores that let in carbon dioxide will also let out water. Okay? And so it's more likely that these guys out here, if we have the Calvin cycle out here, they're going to actually lose water. But C4 plants have evolved a way to bring in carbon dioxide here through special pores, move that carbon dioxide into where the Calvin cycle will occur, and consequently not lose as much water. In the process of this evolved process, in the process of evolving this process, they made a way to more efficiently go through and make glucose from carbon dioxide. So let's see how it works. This process starts right here with a molecule of phosphoenol pyruvate. Phosphoenol pyruvate. When phosphoenol pyruvate combines with carbon dioxide with an enzyme that's found in these plants that I won't hold you responsible for, it makes a 4-carbon intermediate called oxaloacetate. And you've seen oxaloacetate before. There is the 4 part of the C4. Oxaloacetate has 4 carbons. Oxaloacetate gets reduced to malate. That's the reversal of a citric acid cycle reaction. It gets reduced to malate using electrons from NADPH, and you know where those come from. To make malate, malate gets transported into the cell region where the Calvin cycle is occurring. And what does malate do? Malate drops off its carbon dioxide. It drops off its carbon dioxide. So what this system has done is it has shuttled carbon dioxide from the atmosphere into the inner regions of this plant. And in here, this carbon dioxide can more efficiently be used. The carbon dioxide then does exactly what you've already seen. It combines with ribulose 1,5 bisphosphate in a reaction that's catalyzed by Rubisco such that 2 molecules of 3-phosphoglycerates get formed and glucose ultimately gets formed. The difference being that this is occurring within this bundle sheath cell, as you see it labeled here, and not out here next to the atmosphere. The process is efficient, the plant loses less water, and it inserts a couple of steps, but those couple of steps make this overall process work very well. Okay, so that's the difference between a C4 plant and a C3 plant. Questions about that? Yeah. Why aren't all plants C4s? I don't have a good answer for that question, but it's a good question. Okay, but we also have to look at evolutionary processes and say, why do we have duck-billed platypuses? I don't know. At some point along the way, they probably made sense, and they still are able to compete and still able to produce offspring and so forth in a modern world. these work as well. It may be that there's a disadvantage to doing C4 in a non-tropical or a non-dry environment. I don't know the answer to the question, though. Yep. Okay. Well, the last thing I'm going to show you, we might even get out a little bit early today. Let's see, hold on there. That's all the reactions. You don't care about that. All right. The last thing I want to show you is what I tried to show you earlier, and that was what's happening with inefficiency in photorespiration. I told you that Rubisco was a very inefficient enzyme. Okay? Rubisco can take oxygen, and if it takes oxygen instead of carbon dioxide, something interesting and something potentially bad happens, although it's not necessarily bad. If Rubisco binds to oxygen, it will not fix carbon dioxide. Instead, it will combine that oxygen with ribulose 1,5-bisphosphate and make a single molecule of 3-phosphoglycerate, not 2. That's what it makes if it makes carbon dioxide. And in addition, it'll make a 2-carbon molecule called phosphoglycerate. All right? 2-carbon molecule called phosphoglycerate, which will readily fall apart and become glyoxylate. Glyoxylate is toxic. We've talked about it before. And we talked about it with respect to a cycle. And the cycle that we talked about occurred in plants, and it occurred in bacteria. And being able to handle glyoxylate is a very useful thing if cells have a lot of glyoxylate. One of the reasons plants have glyoxylate is because this inefficient cycle actually produces glyoxylate. And the glyoxylate cycle is able to detoxify it by ultimately making the glyoxylate cycle spin. Question? Oh, okay, start over. Back up. That's fine. All right, so let's start over. All right, so let's go back to the beginning here. Talk about Rubisco. All right? So Rubisco is the enzyme that catalyzes normally the reaction that combines carbon dioxide with ribulose 1,5-bisphosphate. This enzyme is inefficient, and it will also bind to oxygen. And when it binds to oxygen, it makes 2 products that are not the same as the 2 products it makes if it binds to carbon dioxide. If it binds to carbon dioxide, it makes 2 molecules of 3-phosphoglycerate. Right? If it binds to oxygen, it makes 1 molecule of 3-phosphoglycerate and 1 molecule of phosphoglycerate. The phosphoglycerate has a couple things happen to it, but it ultimately becomes glyoxylate. Okay, this process of going from phosphoglycerate to glyoxylate happens pretty readily. It's glyoxylate that's problematic for the cell. If glyoxylate accumulates, the cell will die. The cell should have a way of dealing with this glyoxylate that's being produced, and it does. It has the glyoxylate cycle. Now I'm going to remind you, the glyoxylate cycle was like the citric acid cycle, but in the glyoxylate cycle, what happened to glyoxylate? Does anybody remember? It combined with another molecule. What was it? Acetyl-CoA. And when it combined with acetyl-CoA, what did it make? It made malate. Okay? And then we made a second molecule of oxaloacetate as a result of that. In essence, what I've just told you is the plant cell has just detoxified glyoxylate because it's combined glyoxylate with acetyl-CoA, and therefore we have no more glyoxylate sitting around, or the plant has no more glyoxylate sitting around. Okay? So the glyoxylate cycle is mopping up these things that are being produced by this inefficient reaction that's occurring in plants. Does that help? No. That's fine. I don't take it personally. What I would say is look it over, and if you want more help, come see me. How about that? I'd be happy to talk you through it. But the bottom line of this is that I'm trying to give you a sense of two things here. There's really two pathways that are converging. One's the Calvin cycle, and the other's the glyoxylate cycle. The glyoxylate cycle is mopping up a toxic byproduct of the Calvin cycle. And so in this sense, having a glyoxylate cycle makes a lot of sense for plants. It makes a lot of sense. Everybody ready for a song? Okay. This is a song I have that I'm going to have somebody else sing for me because he's much better at it than I am. Anybody know the old song called Scarborough Fair? Are you going to Scarborough Fair? Parsley, sage, rosemary, and thyme. Okay. This one is called Photosynthesis is Divine. All right? And you can sing along with David as he sings this song.
Rubisco acts inefficiently, splitting it into 3 PGs. If the enzyme grabs an O2, it makes glycolate, it is true. The Calvin cycle works in a week, giving plants a sugar in need. So photosynthesis is divine, 'cause it happens all of the time, from dawn to dusk and times in between. Solar panels truly are.
All right, guys. See you tomorrow.